We are here today to discuss the new paradigms in human genetic research that CRISPR technology has enabled. For many, many years, studying genetic variation in humans has only been possible using observational techniques and some very small-scale experimental techniques. But now the development of genome editing technology with CRISPR has enabled completely new approaches to studying genetic variation in humans and other aspects of human biology.
We can't possibly make all of the genetic variants that's possible in the human genome by just looking at natural populations. And this technology allows us to basically go into the genome and make specific edits. Jennifer Doudna, who discovered CRISPR, received an award here last year. And in a year, we've made tremendous advances in how to use CRISPR-Cas9 to do genome editing in non-coding regions of the human genome.
The basic technology for CRISPR-Cas9 is of course a couple of years old already, but now we are really starting to see scaling up of that technology and its use for real biological questions, including both gene knockouts in a very large-scale manner, also more specific edits of the genome, and then different assays both in vitro and in vivo to really probe new questions in disease and basic biology.
We've brought in experts in CRISPR-Cas9 applying the technology to human genomes. And what I hope that people get out of this are understanding of where the technology is today and look at some of the applications that these amazing investigators have used that technology for and really start thinking about how they can be using this technology in their own science.
In some ways, CRISPR-Cas9 enables same kind of precision genome editing approaches to use in humans and human cells that had been previously possible only in model organisms. Even though I think most of CRISPR research in humans is still on the basic research side, there is an increasing interest in clinical applications and in vivo edits, for example, to fix mutations that cause genetic diseases.
It's a paradigm shift. We're able to make mutations that we can't possibly make just by surveying natural populations. Specifically, we can identify places in the non-coding genome that may have more therapeutic potential than natural variation, even though the same region of the genome might be associated with disease.
I hope that people who attend this session will get an overview of what is happening in the field of CRISPR and what are its multiple different applications in different questions, including technology development, developmental biology, basic biology of the human immune system, and then also probing the non-coding genome of humans.